But for the time being, what we want to do now is move on to looking at comparative genomics. So how we can use the Ensembl Genome browser to get information about the comparison of the genes and the genome sequences across the different species that we have in our databases. So in this session, what we want to do is we're going to split the session into two main themes. So firstly, we're going to think about the analyses that we can perform when we're comparing the genes from one species to another. So we firstly going to think about the gene trees that Ensembl computes, and then we're going to think about how we use those gene trees to make homology predictions, so looking at ortholog and paralog relationships. And then in the second part of our talk and the demo, what we're going to be doing is looking at the comparison of the genome sequences from one species to another. So not the genes themselves, but the whole genomes, the sequences where they've been aligned with one another. So this can be performed in a pairwise way, or it can be also a multiple whole genome alignment as well. And we can also think about synteny relationships. So comparative genomics is a really useful tool to help us understand, at least in the context of Ensembl, vertebrate evolution. Obviously, when we're looking at the Ensembl genome species that also have comparative genomics data available, we can think about the evolution of non-vertebrate species as well. So what we can really do is look at the differences at the genome and gene level between species, and we can do some really nice things by predicting gene function based on homologous relationships. And we can look at the distribution of highly conserved regions as well to infer function and importance of specific regions, perhaps. So in Ensembl, we perform comparative analyses by taxa. So firstly, we perform all of our gene tree analyses, homology relationship predictions, and alignments across the vertebrate species within Ensembl. And then for all of the Ensembl genome species, we perform similar analyses on those species related to one another. So the first type is the comparative analysis by taxa. So all the vertebrates against one another, the non-vertebrates, the metazoa against one another, the fungi against one another, and the plants against one another, for example. And then the next thing that we do is we also perform what we call pan-taxonomic comparative analyses. This is where we take representative species from each of the different taxa. So we take some representative vertebrates from Ensembl, and then also some non-vertebrate species from Ensembl metazoa, fungi, plants, protists, and bacteria as well. And then we perform the same analyses as we do for each of the individual taxa. So we look at gene tree constructions and homology relationships, for example, across this representation of the tree of life. So this is a screenshot taken from a gene tab in Anopheles gambiae, so the mosquito vector for malaria. And you can see that the comparative analyses are split into the metazoan compara analyses, so that's the per taxa analysis, and then we have the pan-taxonomic compara, so comparing, in this case, the Anopheles gambiae genes across the tree of life, across Ensembl. So what I want to do now is think about some of the analyses that we perform in Ensembl, and the first one that I'd want to talk about is the gene trees. So the gene trees that we construct are based on protein alignments. So we take a representative protein from each Ensembl gene, and then we perform a process of clustering, blast searching, and multiple alignments to construct the gene tree. Obviously, what I've written here is a very brief overview summary of the sorts of things that we do to construct the gene tree. But if you're interested in the actual methodology, then that's in the documentation here as well. The final thing to do is to reconcile the gene trees against the known species tree. And then the final thing that we do is we take our gene tree and we infer the homology relationships. So we look for the ortholog and paralog relationships between the members of the tree. as I said, one of the things that we do is we infer the homology relationship. So I just want to quickly explain that in a bit more detail. So what I've done is I've just made a cartoon, a quick example of a small branch within a larger gene tree, where we're looking at human, chimp, and mouse genes here. So what we can see is that the two types of homology relationships that we can predict are paralog relationships and ortholog relationships. So orthologs are genes that have emerged through a speciation event. So the speciation events on the tree, you can see, are depicted by these blue nodes, and that's the same in the browser we'll see in a moment. So you can see here there's a speciation node at this position in the tree and at this position in the tree as well. And we can see that those the genes that are connected through that speciation event are ortholog. So you can see that C1 is an ortholog of H1, and equally C2 is an ortholog of H2, and in fact H1 is an ortholog of the mouse gene as well. The paralogs are genes that have emerged through a duplication event. So these are two different genes but within the same organism, obviously where it's been duplicated back in evolutionary time. So we can see that H1 and H2 are paralogs of one another because they're linked through this duplication event. So in this branch of the tree, further back in evolutionary time, there was a duplication event in a common ancestor of both chimpanzee and human, where this gene was duplicated, which gave rise, followed by speciation, which gave rise to the chimpanzee 1 and chimpanzee 2, which are paralogs of one another, and human 1 and human 2, which are paralogs of one another. We can also think of the number of genes that are within this relationship. So we can think of ortholog relationships that are in a simple one-to-one relationship, where there's one gene in the chimp, for example, that's orthologous with one gene in the human. And then we can think of a one-to-many relationship. So we have one gene in the mouse, for example, that's orthologous with two genes in human. So what's obviously happened here is there's been a speciation event between the mouse lineage and the ape lineage on this side. But within the ape lineage, there's been a duplication event, which has meant that there's now two genes in the human that's related through an ortholog relationship with one gene in the mouse. So this is a one-to-many relationship. Obviously, you could imagine a duplication event over in the rodent lineage over here. That means that we would then have a many-to-many relationship. So what I want to do now is jump out to the browser and begin looking at gene trees and homologs, so looking at ortholog and paralog relationships. So if you're following along in the course book, the screenshots start on page 40, but you can also follow along with me as well. So all we need to do is go to Ensembl. I'll open up a new tab in my browser just to navigate to the Ensembl homepage. We're going to look for the human BRCA2 gene to find the gene tree and its ortholog. So in the search bar, we can search for BRCA2 and click go. And then navigate to the gene tab just as we've done before. So from the gene tab, you can see this section in the menu on the left-hand side called Comparative Genomics. So the first thing I'm going to click on is gene tree. And here we can see the gene tree itself. So we've got a summary at the top here with the number of genes that are present in the tree and the number of speciation and duplication nodes, for example. And then underneath we have a tree with a legend underneath that as well. So we can see that the tree is divided into these colored blocks, which represents the different clades, for example. So we've got the primates here in this sort of mauve blue color. Then we've got the Laurastherium mammals in the light blue, placental mammals in green, then reptiles and birds in this sort of peach colored block. what we can see is that the tree is made up of these blue speciation nodes, which, as you can see here, the blue is the speciation nodes, but there are also will be red duplication nodes as well within the tree that we can explore. We can see our gene of interest is highlighted here, BRCA2 human in red, and then we can see that there are orthologs in other species. So we've got the BRCA2 ortholog in gorilla and orangutan and gibbon. What you can actually see is that there are a number of species that are hidden underneath these collapsed funnels. So these grey funnels represent a collapsed branch of the gene tree, just to show a small section of the tree by default. But if you want to expand the tree, all you need to do is click on the grey funnel, and then you can click on the link in the pop-up window that says expand this subtree. So once you click on that, you can see now all of the old world monkeys are now also being shown within the primate part of this tree. You can do the same thing for the placental mammals as well, for example. So there's lots of, you can customize this tree to show as much or as little as you like. Over on the right-hand side of the tree, we can see information about the protein alignments that were used to actually build the tree. So if we scroll down, we can see what these green and white blocks represent. So when we're looking at an expanded alignment, a white is a gap in the alignment and green is a line sequence. So what we can see here is that where we're looking at just a protein that represents a single gene, so here we've got the human BRCA2 protein here. Where it's green, we have a line sequence with other species, and the white is where we have gaps in the alignment. Where we have trees that are hidden by these funnels, so these branches of trees that are hidden by funnels, we have this protein representation that's filled up with light green, dark green, and white. So what we can see here is that it's just a single protein that's meant to represent all of the bony fishes in this case. So we can see that the white is between 0 and 33% aligned sequence. The light green, which you can see here, for example, and here, is between 33 and 66% aligned sequence. And the dark green is where we have between 66 and 100% aligned sequence, but for all of the orthologs in this bony fishes branch, for example. If you want to download this tree, what you can do is you can export this image using the picture icon in the blue bar at the top, or you can download the data from this tree using the box with the arrow pointing into it. So if you click on the box with the arrow, you'll open up a pop-up window that you can use to export the tree in one of a number of different formats. So you can download the gene tree in clustral format, FASTA format, Mega format, MSF, NEWIC, NHX, OrthoXML. These are all different ways of representing the gene tree and the branches and the branch lengths, for example. So depending on your downstream application, will determine the type of the gene tree format that you select. So all you need to do is click your format of interest or select it from the drop-down list and then click download. So one of the things, I'll close this window now just to get rid of the pop-up window. So one of the questions that's just come in is, can a phylogeny tree be made here using your own DNA sequence or a submitted DNA sequence? So I can see Mihal's just answered that question, but the important thing to remember when we're looking at all of the comparative genomics data in Ensembl is that this is pre-computed data based on the gene sequences and the genome sequences for the species we have in Ensembl. So you can't use Ensembl for performing your own alignments or constructing your own trees. This is just a way of retrieving the pre-computed analyses that we do within Ensembl. So there are many other tools that you can use for alignments and gene tree construction, but Ensembl is not one of them. The other thing that I wanted to look at in this portion of the demo is again from the menu on the left-hand side. It's called Orthologs in the menu. So if you click on Orthologs, What we'll see at the very top of the page is a summary of the ortholog relationships across a number of species. So obviously we've got human, BRCA2 as our gene of interest. And then as we scroll down, we can see that, for example, if we look across the 26 primate species in Ensembl, there are 25 with a 1-to-1 ortholog, and there's actually one without an ortholog prediction as well. And then that's broken down by the different species sets you can see here. Underneath the summary table, we actually have a larger table that shows us the orthologs in all of the different species. So, for example, the Algerian mouse has a 1-to-1 ortholog with the human BRCA2 gene, and this is the Algerian mouse BRCA2 ortholog. So you could click on this ID for this gene, and that would take you to the Algerian mouse BRCA2 gene, which you could continue exploring as well. If you want to filter this table and search for a species of interest, you can use the filter option in the top right-hand corner. So I can just begin typing mouse, for example. Obviously, we've got the Algerian mouse, mouse lemur. So it's not, the text search is just based on, it's just a text search rather than using any sort of knowledge about the relationships of species. So as long as the word mouse occurs in the name, it will come back to you. So mouse lemur as well. But then we also have mus musculus. So this is the mouse that's used as a common, the mouse species that's used as a model organism. And you can see here, this is the 1-to-1 relationship that we have, and this is the mouse BRCA2 gene. In the table, you can see what we call the target percentage ID and the query percentage ID, which tell you the amount of a sequence alignment between the two orthologs. And then we have gene order conservation score, whole genome alignment coverage, and also the confidence in the ortholog prediction. The final thing that you can do, if you click on view sequence alignments, if I use the mouse as an example, you can actually view an alignment between the human and, in this case, the mouse cDNA or protein sequences using the pop-up window. So if I click on view cDNA alignment as an example, it's just taking a moment to load for me. So just while this page loads, it's just loaded for me, but just while it loads, there's a question in the chat box, and it says, it's from Sybil. All of this is based on nucleotide sequences, correct? Is there a similar approach with the amino acid sequences? So the gene trees are actually based on protein alignment. So we take a representative protein from each gene, and we actually perform alignments on the protein sequences from the representative protein to build the gene trees. But then we do provide you with the cDNA alignments, for example, between the orthologs. And then in the next section of this module, I'll be explaining to you about the whole genome alignments, which is obviously based on nucleotide sequence alignments. If you want to get more information about protein alignments, I'm not sure of the best resource, but within the EBI, you could probably go to UniProt or use the Clustal Omega tool through the EBI pages that might be able to help you with the protein alignments. So on this page, we can see that we have, that's okay. On the page, we can see we have the ortholog alignment in Clustal W format. So at the top here, we have a summary. So we've got the human gene and the mouse gene. We've got the length and the percentage identity between the two sequences. And then we have the sequence alignment in Clustal W format. So the ENSP This is the cDNA sequence for the human protein, and then the ENS MUSP, this is the cDNA sequence for the mouse protein. Obviously, the stars and the different characters on the third row represent the alignment the alignment between the two sequences. You can download this alignment if you click on the blue download homology button in the top here and underneath Ortholog alignment title. Good. So the final thing that we're going to do today in our session is to quickly, is to also look at the whole genome alignment. So what we've been focusing on so far is the comparison of the genes across species, but now we want to think about whole genome alignments. So whole genome alignments are really useful to help us to identify highly conserved regions across species. These are sequences that are likely to have evolved slowly, and they're also very likely to be functional as well. So they can include both coding and non-coding sequences, and they're really useful to help, at least in Ensembl, to trouble spot gene predictions and to also to define syntenic regions as well. So there's two types of whole genome alignments that we perform in Ensembl, pairwise and multiple. So the pairwise alignments are performed with BLAST-Z, which is older, or more recently we've been using LastZNet. And we perform the pairwise alignments against, obviously, paired species against each other. So for human, we align the human genome against every other species in Ensembl. And then for model organisms, we align the genomes to related species. So for example, we align the mouse genome to other related rodents. Agricultural mammals, we align to each other as well. And we also perhaps align species sets, so the fish against one another. So if you are interested in the different pairwise alignments that we perform, you can get the information through the documentation that's linked here. So the multiple alignments are performed on predefined species sets. So for example, the EPO analysis, the Enredo, Pecan, Orpheus multiple genome alignment pipeline performs whole genome alignments across the species set, so the fish, or the sauropsids, or the eutherian mammals, or the primates, for example. So in Ensembl, we have species sets, so the primates forms one species set, for example, and then the EPO pipeline will perform the multiple whole genome alignment across that species set. We also perform an EPO extended analysis across even larger sets of species that are a bit more distantly related. Then we also have the Mercator-Pecan pipeline analysis that performs whole genome alignments across the amniotes, so the mammals and the birds essentially, so the amniotic vertebrates in Ensembl. And then we also use cactus as a whole genome, multiple whole genome alignment across the Murinae species, so the mouse strains and other related mouse species. So what I want to do in our final demo of the day is to look at a region of the human genome which contains the HOXD cluster, and then we're going to find conserved regions and alignments to this region of the genome. So in the course book, we're starting on page 43, but you can also follow along with me as well. Again, I'll just open up a new tab in Ensembl. So what we want to do is we want to search now for our region of interest. So we have a genomic region that we want to find the alignments across different species for. So I'm going to search for human, a region in human, and I'm going to put in my location of my genomic coordinates. So we're looking at a region on chromosome 2 that contains the HOXD cluster. And then the start coordinate is 176.087.000. And then a hyphen, and then our end coordinate is 176.202.000. And I will put that in the chat box for you as well. And then when you've put in your coordinates, you can click go. and that will take you to the location tab. So again, we've seen the location tab before. This time we're on a region on human chromosome 2 that contains the HOXD cluster. So just while the page is loading, you can even see this region of the genome where these HOXD genes are located. Many of you probably have studied or know about the HOXD cluster of genes, very important in embryological development and limb formation, for example. So now the page is loaded. So what I'm going to do is I'm going to click on configure this page to add some comparative genomics tracks to our location view. And then in the pop-up window, I'm just going to scroll down a little bit to find this subcategory in the menu on the left called Comparative Genomics. And here you can see all of the comparative genomics tracks that we have available. So firstly, we have a set of tracks that will display the multiple alignments across the different species sets, as I explained to you before. So we have the EPO extended analysis across the 111 eutherian mammals represented in Ensembl, or the 13 closely related primates, for example, using the EPO analysis. Then we also have conservation regions. So these are conserved regions that are calculated based on these multiple alignments. So for example, we have the constrained elements computed from the 111 eutherian mammals, the multiple alignments across the 111 eutherian mammals. So we already are seeing the constrained elements, and then we'll also look at the conservation score as well. So I'll click on the empty box to turn on the conservation score for the 111 eutherian mammals as well. You can also see underneath that we have tracks that will show us the pairwise whole genome alignments from the human region to all of these other species as well. So I will just select a couple as an example. I will choose perhaps cat here as an example, and I'll select the format as normal. I'll also select cod, maybe a bit more distantly related. And then I'll also select chimpanzee, so a closely related primate. So I've just selected 3, trying to pick sort of a closely related primate, and then a mammal, and then even a more distantly related fish as well. So when you're happy, you can save and close. And the page will just reload with the new data tracks that we have selected. Good. So we can see now that these are the new tracks that we've added. So this is the 111 way GERP scores. So this is the conservation scores across the multiple whole genome alignment across the 111 eutherian mammals. So what we have here is an axis here going from 2.84 to minus 2.68. So what we have here are the positive scores that represent conservation across the 111 species in this alignment. And the negative scores represents a lack of alignment, a lack of conservation across at this position across those 111 species. So what we can see here is that there are regions of positive score. So we'd say that this region is highly conserved, and that's where we infer these conserved blocks from. So this is the track showing us the 111 way GERP elements. So these are the constrained elements, the conserved blocks that we infer from these regions of positive score in the conservation score. You can see here, for example, there's a region of negative score, so a lack of conservation across these 111 species, and you can see the corresponding lack of conservation here. What I find quite interesting is if you, if you're interested in sort of alignment and comparison across species, you can actually notice that these conservation blocks, these conserved regions, actually very closely match up with the coding sequences of these HOXD genes, obviously indicating their functional importance across these 111 eutherian mammals. if I scroll even further down, you can also see the alignments that have been put at the bottom for me. So we're looking at this region in human, and then we have the alignment against each of these 3 species that I selected as well. So human against chimpanzee, the pink block represents aligned sequence. So we can see here there's 100% aligned sequence between the human and the chimpanzee region here. Then we have the cat, obviously a mammalian species, but a bit more distantly related. So we have some white blocks that represent the, there's no alignment in these regions. And then the cod, we can see that we have these pink blocks, but lots of unconserved regions as well, represented by the white regions. Good. Okay, so the very final thing that I want to show you is through the menu on the left-hand side. So at the moment, we are looking at what we call the region in detail view. This is what we've been exploring a lot through the location tab. But there's also a section here called Comparative Genomics. So the first thing I'm going to click on the menu on the left is the alignments text option. So what I can do now is I can retrieve an alignment of this human region against another species of interest. So all I have to do is click on this blue select an alignment button. And that will open up a pop-up window that we call the species selector. So you can either begin typing a species of interest into the window here, into the text box to select your species of interest, maybe mouse. And then you can check the box to choose your species of interest. Or you can view all alignments and you can choose using these options here. So if you want to view the rats and mice, for example, again, you can select from here, or the lagomorphs, pika and rabbit, and other rodents. So you can either type in your species name, or you can select using the clicking options. So I've selected the mouse, which is the mouse reference, the CL57 Black 6 mouse strain used as a model organism. That's the reference mouse genome sequence. So I can then click apply. And you can see that now the human region that we have selected in our initial search has been aligned to these different blocks across the mouse genome. So there's an alignment block of around 115,000 base pairs from human to this region in the mouse. And then there's also these other smaller alignment blocks of around about 500 base pairs. So what I'm going to do is click on block 1, which is the alignment between the large sequence within our region of interest. And that will allow me to scroll down, and I can actually see the alignment between the human and the mouse region. So this is just a preview. If I click display full alignment, I can actually see the full alignment between the human and the mouse regions. And then in red, you should be able to see these are in red, we have highlighted the coding sequences. So you can see actually the coding sequences are very well conserved, with perhaps the gaps coming in the non-coding regions. If you want to download the alignment, you can do that using the blue button at the top as well. Finally, you can click on region comparison from the menu on the left. And this allows you to compare the region in detail page across 2 species. So at the moment, we're looking at the human region in detail. So we've got the chromosomal overview, we've got the 1 megabase overview, and we've got the region in detail view with the tracks. But if we click on the blue select species or region button, again, we can choose our species of interest from the species selector menu and click apply. And now we can see the region in detail view side by side for these 2 species. So we've got the chromosomal overview for human and mouse, so Homo sapien chromosome 2 and mus musculus chromosome 2. Then we've got the 1 megabase overview between the human and mouse side by side that you can compare. And then you can also see the aligned image between the human and the mouse region. So we can see the HOXD genes here in human that correspond with the HOXD genes in mice. Very good. Okay, so I think that takes me to the end of the demo. So we just have around 15 minutes left together. So what I'd like to do is give you an opportunity to have a go at the exercises for this module. If you go to the question book, the questions start on page 12. So we can see here we have a question looking at orthologs and paralogs of the human BRAF gene, looking at cow orthologs and also some whole genome alignments as well.